So we are a couple of minutes past the hour, so I think it's good time to get started. So good morning, good afternoon, or good night, wherever you are. Welcome to Speaking of Oats. My name is Pablo Oliveira. I'm director of Oat Global. Speaking of Oat, it's a partnership between the Oat newsletter and Oat Global. Charlene White, editor of the Oat newsletter, is with us today and will be handling the question and answers after the seminar. Oat Global is a public-private partnership that aims to coordinate and connect all researchers and oat extension professionals worldwide with each other and with individuals along the entire oat value chain. We are glad you're here today. But before I introduce today's speakers, I want to take a moment to remind everyone that you can use the Q&A widget to ask questions throughout the presentation. Please use the Q&A widget and not the chat function for questions, since this preserves the question for later access. Today's session is being recorded and will be made available later via YouTube. Today we have a group of speakers from Europe that will present the CROPDIVA and FUGE projects. CROPDIVA, or Climate Resilient Orphan Crop for Increased Diversity in Agriculture, it's a multinational project that works on the promotion of 6 underutilized crops, including oats, and the creation of new value chains based on these selected crops. CROPDIVA has a multidisciplinary approach that integrates different disciplines like agronomy, ecology, breeding, food and feed science, economy, data science, social science, and marketing. And FUGE, it's also a collaborative project funded by the German Federal Agency for Agriculture and Food that it focuses on the development of genetic tools for more efficient oat breeding and monitoring fusarium species on oats. So today we will have a longer webinar. As we will be presenting these 2 projects, today's webinar will be extended for an extra half an hour. So thank you to all the speakers for being here today. And now I will hand the floor to Matthias Herrmann to start the presentation. So Matthias, the floor is yours.
So thank you, Dr. Firbo. I'm honored to introduce into the project monitoring of Fusarium species. Moment. So everyone can see this. Monitoring of Fusarium species on oats and development of genomic tools for more efficient oat breeding. This is a German project which has been started in December 2020 and will be finished, I think, in 23, but we try to get a prolongation till 24. So we do have in that project 5 scientific partners. These are the GKI, which I'm working with in, and then the IPK, Martin Mascher, the Plant Genome and Systems Biology Center in Munich, then the University of Göttingen, the University of Gießen. And we do have 3 breeding partners in that consortium, the KWS, with Klaus Oldach, the Nordsaat with Steffen Beuch, and the Saatzuchtbauer with Sophie Brotführer. So this funding is given by the Federal Ministry of Food and Agriculture, and the project is managed by the Federal Office for Agriculture and Food and by the German Federation of Plant Innovation. So we do have 4 work packages. In the work package 1, work package 1 is mainly processed by Charlotte Rodemann and supervised by Professor Andreas von Tiedemann, University of Göttingen, and they looking for the monitoring or occurrence of Fusarium species and mycotoxins on oats across Germany. And a second part of this work package is the detoxification. We will get some results about that monitoring today, later on. In the second work package, the cross resistance to Fusarium poe, Fusarium sporotrichoides and Fusarium graminearum are the first main part. This is the main question is how Is there a general resistance against these three species? And is it necessary for the breeder to install or to arrange separated nurseries for selection of these against these three species? Involved are the three breeding companies, and we do have 25 have selected 25 cultivars for that task, and these 25 lines are evaluated at 4 sites in during the last 2 years, and because Fusarium poe is producing nivalenol, and there's no ELISA kit available for that. So we quantify not the mycotoxins directly, but we quantify the Fusarium DNA via qPCR. This is done by Charlotte Rodemann in Goettingen. And well, the results are on the way, and we will see what will come. So the second part in that work package 2 is focused on trichomes in oats as a possible resistance factor, and this will be presented today by Selma Schurack. The work package 3 is led by Martin Mascher and Manuel Spannagel, and is targeted to sequencing and annotation of oat genomes by the project FUGE. We has enabled the sequencing and annotation of 3 oat genomes of modern cultivars, namely Delphine and Lion and Rhapsody. And this is embedded into the International Oat Project, Pan-Oat Project. Martin Mascher and Nick Tinker are leading this project. Martin already have given a talk about the pan genome within Speaking of Oats already, and also presented pan-oat results in Perth on the International Oat Conference. Results are nearly complete and enabling now a more efficient analysis of structural variation and the development of specific marker assays. In work package 4, genomic selection is started with a first step or first cycle. This project is initiated by Matthias Frisch, University of Gießen, and for this we have developed 250 oat lines or oat progenies from 25 crossings and out of 15 parental lines. And we have already tested the yield of these yield and agronomic traits of these 250 oat progenies on 5 sites, and Fusarium resistance meanwhile on 4 sites. Results are not complete, but analysis and results are on the way, and also genotyping for that purpose is on the way, and is made by or organized by Martin Mascher and IPK. So we are curious about the results which will come next. So that was the introduction into this big project, and I would hand over. I would thank you all, and would like to give over to the next speaker, to Charlotte Rodemann. She will give or present her results about the monitoring of that project. Thank you. So let's stop.
Yeah, thank you for your introduction, Matthias. And as you already mentioned, I'm going to talk a bit about the monitoring aspect of our FUGE project. Because it's embedded in it. It's one part of 4 parts of the FUGE project, and so I'm just going to present you some recent results and give you a little overview. So we know that the consumption of oat-based products has recently increased in Germany or over the last few years. However, the production of oats has only increased slowly, and so we just want to, one aim of our project is also to give a better overview about what can be done with oat in Germany. And we, however, don't know much about what is going on with Fusarium in Germany. And we have also recently some investigations that showed that actually some of the commercially available oat products in Germany are contaminated with T2 and HT2 and also exceed the limits that have been set by the EFSA. So therefore, we doing this monitoring to understand what's going on in Germany, what species are relevant, and what other mycotoxins could be present in the German oats. So therefore, we conduct this monitoring process. We determine the Fusarium species. Therefore, we collect samples from different locations around harvest, and we mainly focus on the 3 spring oat cultivars, Max, Apollon, and Lion, which belong to the most used or cultivated cultivars in Germany, and we identify our Fusarium species morphological, but also with molecular tools, and we perform our analysis for the mycotoxins via HPLC. And to have a better understanding, we also collect agronomical and meteorological data. So we collect, for example, the crop rotation, but also data on for rainfall or temperature during the cropping season. Here's a workflow overview. So we have a set of kernels which we surface sterilize, and then we isolate from these infected kernels, cultivate the Fusarium species, and identify them via macroscopic parts, but also via microscopy. And then we later on use our molecular tools like species-specific primers, also the sequencing of taxonomic informative regions to identify the occurring Fusarium species. I'm now going to share with you the results from the year 2021. We already started in 2020, but I'm mainly focusing on the last year. Sadly, in the last year, we had only 10 locations across Germany. We talked to some of our partners, and they somehow said that at some locations we had heavy rainfalls, and the fields were just not, they couldn't harvest them. And but overall, we have to say that last year was there were high incidences of Fusarium. We had an average about 36.3%. We couldn't see any differences between the cultivars. They were more or less equally infected, but we saw a high variability between the locations, as you could also see here. I just wanted to roughly show to you how the incidences or the number of infected kernels were. We had some locations that were really highly infected, and some where it was definitely less. If we look now at the species spectrum we could found in the last year, then we can see that we have lots of Fusarium abenaceum, Fusarium tricinctum, and also Fusarium poe. In some other minor species, we couldn't only find a little bit of Colmorum and Fusarium graminearum, which are known producers of deoxynivalenol. But we also found Fusarium sporophytoides and Fusarium equiseti. And I think what's interesting to some of you is that we only find very, very low frequencies of Fusarium lanzetii in Germany. I now included on the next slide our results from the year before. What I have to mention for the background is we had 2 more locations in 2020, and in general, the Fusarium incidence in 2020 was way lower than in 2021. And also we see here only I included on the Fusarium lanzetii and some other species under weitere Fusarium species, which are only Fusarium species which occurred under less than 1% on their own. And we can see we have not that much lanzetii in Germany. We could see in the year 2020 that we had much more Fusarium poe compared to 2021. But if you look at the 3 main species that occurred in Germany, it's still Fusarium abenaceum, Fusarium tricinctum, and Fusarium poe. Just the frequency changed. Yeah, no, I just want to mention some things about the mycotoxins, like a summary. It's also only for the year 2021. Yeah, we could find in more than 50% samples T2s, like sometimes also in quite elevated levels. Same goes for HT2, so we've only found it in 30% of the samples, but in some the amount we found was elevated. However, DON was only could only be found in 30% of overall samples, and we found no free A-DON, 15A-DON, or glucosylated DON, and also no nivalenol. However, when we look at other mycotoxins, for example, the initians, we could found A1, B, and B1 in up to 90% of the samples, and sometimes also in quite high levels. Yeah, just to give a general summary and the outlook, I think what we can take from this is that in Germany we have a lot of Abenaceum, Zinctum, and Poe. Also, if you look over all about 2 years, the last 2 years, those three were the most present ones, even though the frequencies change between the years. And I think what is also very important is that we have a lot of T2 and LT2, and I think that's something we should always consider. And we want to continue this monitoring for the year 2022, and we are also looking into including maybe samples from mills to get a better overview about what's going on. And I would like to thank you all of our partners who kindly sent me the samples, and I think I want to thank you for your attention, and will now hand over to Selma.
Okay, one moment. Okay, I hope it works. So yeah, thanks, Charlotte. Yes. So I will be the last presenter of the Fusarium project. So in oats, a few morphological features that influence Fusarium resistance are already known, such as, for example, plant height and enter retention. However, the resistance components have not been fully elucidated yet. So here I will give a brief report on our preliminary results on the possible roles of trichomes during Fusarium infection of oats. And if we take a closer look at the beginning of the infection process in the panicle, we see that the primary interface is mostly the outer floral parts where the Fusarium spores germinate. And the infection typically starts here at the apex, on the ventral side, so mostly on the palea, and the proliferation of the fungus then proceeds to the base and to the dorsal side. And this is also visible here on the microscopic picture taken 1 day after inoculation, where we can see the fungal hyphae growing on the apex of the palea towards the base. And consequently, it is quite conceivable that chemical or anatomical features at the husks, as for example trichomes, might play an important role during Fusarium infection. So what actually are trichomes? Trichomes are unicellular or multicellular appendages that are formed exclusively by epidermal cells on the surface of plant organs. And they are also referred to as plant hairs, and they can have various sizes and shapes. And in other hosts, for example, barley and maize of Fusarium, trichomes have been found to be preferred entry sites. And this picture here shows the Fusarium hyphae in green during the penetration and the colonization of a trichome on the lemma of barley. And here we can see a macro trichome with the hyphae growing on the base and in the middle of the trichome. However, there are also some studies where opposite relationships have been found. So for example, in wheat, Fusarium head blight resistance plants were having more trichomes. So this led us to the question whether genetic diversity in trichome size and density could affect Fusarium infection in oat. And to investigate this, the panel from the cross-resistance trial that was mentioned by Matthias in the introduction was used. And this panel features comparable growth heights and flowering time, as these are factors that influence fusarium resistance, and we wanted to limit the influence of these. And the plants were grown in the field and in the greenhouse as well. And at anthesis, the lemma and palea were collected and fixed in ethanol to remove the chlorophyll. And then using a fluorescence microscope, the trichomes on the surface of lemma and palea were visualized. So here are two extreme types as an example of how much the trichome density actually differs across our oat lines. So here on the left, we have an oat line with a lot of trichomes in the palea. And in contrast here, we have another oat line with significantly less trichomes. And yeah, this was then quantified, so classified by size in big, medium, and small. The trichome density was evaluated in the 25 oat lines. And here are the combined results for the lemma and the palea. And here again, it shows that there's clear differences in the number and also in the size composition of the trichomes in our investigated oat lines. So, for example, some have a really reduced number of trichomes or almost no large trichomes. And in contrast, for example, here we have an oat line with many big trichomes, or just in general high trichome density. So, of course, the next question was whether these differences in trichomes are related to resistance differences. And for this, a trichome index based on the trichome size and density was calculated. And if we compare this index with the Fusarium graminearum DNA content, we actually found positive correlation of around 0.5. This result is based only on the data from our first trial year, hence should still be considered as preliminary. So to conclude, we can say that at last in our trial, oat lines with fewer trichomes generally had a higher resistance to Fusarium. However, if you want to use this characteristic for breeding purposes, you should of course bear in mind that fewer trichomes are associated with greater susceptibility to insect pests, for example. And further work will first of all aim at validating the results in the second year. And we would also like to understand what actually is the basis for this observed relationship. And for this, microscopic studies in the interaction of Fusarium hyphae with trichomes in selected oat varieties are already underway. So yeah, with this, I would like to thank all the project partners, especially my colleagues at the JKI and also Charlotte, who provided the Fusarium graminearum DNA data. And thank you all for your attention.
Great. Thank you so much, Matthias, Charlotte, and Selma as well. Yeah, the Fusarium work obviously is very, very important. And it's great to see that's being studied now more in Europe too. We do have, maybe we can pause now before we get into CROPDIVA and take about 10 minutes for questions if anybody has a question about the FUGE project. Susanne did have a question here asking what the limits of guidance values are for T2 and HT2.
Well, it was less about whether, not what the guidance values are, because you were talking about limits. Are those legal limits? Because I'm only aware of guidance values.
Yeah, there are more recommendations.
Yeah, right.
So maybe I mixed something up, but to this day there are recommendations, but I think they're working on setting a limit.
Yeah, that's now a couple of years that I'm working on that. So I didn't miss something because I was when I saw that slide with the limiter. That's interesting. Okay, thank you. The other question, since I'm already talking, I had was because you mentioned, Charlotte, that you had very little Fusarium langsethia, and you did both the morphological seed health test and the molecular identification. But did you also do a qPCR, like a quantitative PCR? Because we also underestimated Fusarium langsethia in oats when we did a monitoring in Switzerland. And was surprised because we had so much T2H, and then we did a quantitative PCR, and then we saw that we underestimated that tremendously.
Yeah, we want to include it. We have the samples, and I think we also have some results, but I didn't include them. And we also want to do this for the other samples. But I was aware of your publication, so therefore we also wanted to do the qPCR to figure out if we are underestimating this effect.
Mm-hmm. Okay, thanks.
Great, thanks. I see that Osman Bjornstad has his hand up. Annie, can we allow Osman to speak, please? Otherwise, questions can be put into the Q&A.
Let me see if that is something I can do. Yes, go ahead, Osmond.
Yes. You hear me now?
Yes.
Yes.
Yeah.
Oh.
Thank you very much. Very interesting project. I have a simple question. Is the occurrence of T2 and so on, is that how much of that is in the hulls and how much is it left after dehulling? Obviously, in the grains that you spoke about, you know, it was in the actual product, but I just wonder how much is in the. I never seen data about that. So.
So for the monitoring, we didn't dehull the kernels because we don't have the possibility. For this recent investigation that was actually done on like basically the products that you could buy in the market. So they were already dehulled, that were flakes. And that was an independent group of basically product testers who I think they worked together with a lab and then they analyzed it. So it's not related to our work.
Okay.
Yeah, and with dehulling and this difference between content in dehulled and hulled grains is not a question in FUGE or in that project. So we didn't look for that, but it's known that a major part is in the hulls and get lost or get away if you dehull.
Thanks.
I ask one.
Sorry, go ahead.
Yeah, the reason is that we also done such a project in Norway, and I mean the general impression is that the Langsethia at least gives very little visible symptoms, whereas it does produce. If you inoculate quite heavily, it does produce T2 and HT2. So, and I suppose also by you can measure it by qPCR, but actually, is there? So there is no measure yet of any of the effect of dehulling there, because I mean, in Graminearum you can see the whole grain destroyed quite quickly. But how is that actually in these samples then? Do you see it empty grains, black grains? You know, do you see all that you?
Yes, yes.
With the Langsethia?
Not in Langsethia, not so much, I think. But it is not involved in that project. Not so. Besides the monitoring.
But in the samples where you find T2 and HT2, are the grains frequently decayed like in Graminearum, or are they not?
I don't know. Charlotte, you know?
Normally we don't have that much decades. No, I cannot. Sometimes it depends, obviously. Only sometimes, not that often, and also not that much. It's like a few, maybe.
Okay, thank you.
Yeah, this is one of the big questions, of course, is how do symptoms correlate to actual toxins present? Excuse me. So that's yeah, something to be really careful about. There's a nice segue here actually to Sissy Zechner has a question to Selma. Did you also include naked oats in your studies? And they have generally, unless you're using one of Vern Burroughs' lines, they have many more hairs.
Yeah, no, we didn't really include naked oats, but these hairs, are they not kernel hairs?
Right, they'd be hairs on the groat, and that actually was one of my questions too. Was I know you were looking at the hull, the lemma and palea, but what about the groat itself? Is anyone looking at those hairs?
No, for now we are focusing on the lemma and palea, because that's where the spores land on probably, and that's also what we found in literature, which has been investigated before. Yeah.
Of course, I suppose the naked oats also, they do still have lemmas and paleas, they just get left in the field. So yeah, that might be something somebody could follow up on at some point with some further work. Okay, and I'll just have one more quick question here, I guess, before we get into CROPDIVA. So Jennifer Mitchell-Fetch is wondering, what was the range in parts per million of T2 that you found?
I would have to look that up, so I cannot remember that like out of now from now. So yeah.
So you have homework now. Okay, that's great. We can follow up with a lot of these things later too. And if others have more questions about this project, there will still be more time after we hear the next set of presentations. So I guess we'll be moving on to the CROPDIVA project now. So who's starting that off? Is that Geert? Are you starting that off?
Yes.
There we go. Over to you then. Thanks, everybody.
Okay, good morning, good evening, good afternoon. Many thanks that we have the floor to present, I think, a very nice project, CROPDIVA. And CROPDIVA is, of course, an abbreviation, and it means climate resilient orphan crops for increased diversity in agriculture. And when we look to the main general goal of CROPDIVA, that is that we will reinforce agrobiodiversity on different levels, and we will do that by combining agrobiosense, which include crop husbandry, of course, but also ecological and genetic research. But we will also focus on socio-economic studies and product development. It's a holistic and inclusive approach that we will use, and that means that we must have a lot of experience and different competencies in our consortium. CROPDIVA, the CROPDIVA concept is built around 6 what we call underutilized crops. We work with oat, triticale, buckwheat, whole-nest barley, lupins, and faba beans. And yeah, of course, the choice of these crops are not at random. We are thinking a lot about it, and we select the 6 crops because of their wide genetic diversity, but also they have specific breeding pools, which availability to adapt to different growing conditions. Also, they have a certain flexibility to fit rather easy in diverse crop rotation systems. And they have also, because of the wide genetic diversity, a large adaptation to different growing conditions. They have also nice and interesting nutritional properties. And if we look to the economics and the technological value also in that perspective, they are very promising. We have defined key elements to obtain our deliverables. So we construct a broad and diverse gene pool for each selected crop. We install also a multi-location experimental field network. We will do, and we have already done, a lot of phenomics and genomics. We will define biodiversity indicators. Also, we will look to develop mathematical and data mining algorithms, mapping ecosystem services, and last very important key element is that we also will develop a decision support system for stakeholders in respect to agro biodiversity. So we have many tasks, and that means that we can lay on a lot of partners. In total, 28 partners are included in the project, spread over 11 countries in Europe. And we have a budget of approximately 6 million euro. The coordination is in the hands of our university, Ghent University. And maybe I can also add that we have an external advisory board, and that means that even more countries are involved in our project. So our projects belong to the Horizon 2020 goals. And if, of course, it's financed by the European Commission. So on the map, you see all the countries that are involved: United Kingdom, Denmark, Belgium, of course, the Netherlands, Germany, Poland, Czechia, Switzerland, Austria, Italy, Serbia, Spain, and now you will mention, of course, France is not included, but France is a partner because we have a French partner in our external advisory board. When we look to the structure of our project, in total we have 7 work packages. The first work package is about the interactive information platform. So we will create a database not only based on our results, but also we will look to historical results. We will look to results obtained by other projects, of course, realized in the same domain. Work package 2, it's all about germplasm diversity, breeding, testing, breeding. We will in that work package we will do a lot of phenotypical but also genotypical work. That's work package 2. Work package 3 is about cropping systems, and in work package 3 we will emphasize on intercropping with the 6 already mentioned crops. Work package 4 is about new food, feed and non-food products. And so in work package 4 we will look to technological aspects of the harvest products of our selected crop. And we will look what is possible to develop new food and feed and even non-food products. In work package 5, it's all about socio-economics. So we will look to regional value chains, marketing strategies, but also very important producer-consumer links. And work package 6 is the communication, disseminations and exploitations of our results. Work package 7 is just about coordination and project management. So to conclude, I can say that CROPDIVA is challenge and demand driven, creating different levels of diversity, taking into account different geographic and regional levels, and also it's a very interdisciplinary project and has a multi-actor approach. So that's all what I want to say as introduction. And now I will give the floor to Hermann, and Hermann will present the first results that we have obtained about oats during the first project year. So thank you very much for your attention.
I think Steffen is next, right?
Oh, sorry, Steffen. Yeah, sorry. I go a little bit too fast. Sorry.
Never mind. Okay, so. Okay, I hope that everyone can see it. Before presenting the first results, I would like to give you an introduction into a part of our oat-based trial results, because we have, speaking of oat seminar, and the idea behind these, what we call facultative oat trials, was created a couple of years ago. And it has, of course, to do with what is not very positive to all of us. It's climate change. The climate change is going on. As all of you know, we have a dramatic increase of temperature that have never seen before 2000 years ago. If you look back here in the IPCC report from 2021, that was never given. And in more detail on the right side, especially between 1850 and 2020, there's a really dramatic increase of nearly 1.2 degrees. This is the global surface temperature as measured. And also as a consequence here for Germany, but we could also take another country, but Germany is more common to me. These are scenarios based on the temperature, surface temperature here in the left picture, 1971 to 2000, and simulation, future calculated with the help of a model project on climate simulation for 2033 and 2060, and to the end of the century, which will not be of interest to most of us, but nevertheless in the middle, this will be, we all will be faced with that. And you can see that we will see with a high probability, a high increase in temperature in the surface, which will of course change the supply with food, will change our cropping systems, will maybe change also our crops. And one idea behind that related to oats, because we all know that oat needs more cool climate, more moist climate, and does not like mostly this very hot or very dry conditions that are just coming up with climate change the next decades. The consequences to outcropping was given on the International Oat Conference in 2008 in Minnesota by Mark Seeley. And climate change for oats, he said, can be separated into direct and indirect effects that can be measured. Direct effects may be skewed to mid-latitudes, a longer growing season, which can be also positive, and asymmetric temperature change with higher minimum temperatures, especially in the night, and more degree days. Carbon dioxide fertilization, maybe also positive, I don't know. More biomass, water use efficiency is improved. Moisture stress, seen both as saturated soils and also drought, buffered, of course, by the soil type and the tile drainage, and higher dew points that may favor also new or known crop diseases that are just coming up. And for the indirect effects, we expect milder winters, which favors availability of pests, population turnover rates of plant pests, more rapid degradation of crop residues. That means a more rapid turnover of nutrients in the soil. We see the risk of leaching of nutrients, erosion can come up, change interactions with the surrounding ecosystems, irrigation resources probably will decline, crop rotations will change, they are already on a run to change, and everything will become more expensive. So this is, of course, the negative side of it. So many challenges what are coming up. So this is not only directed to oats, but of course also oats are also faced with that. So what does it mean? If we have a look to Europe, and this is a European project, CROPDIVA, we can also define mega environments for the outgrowing in Europe. That was given by myself on a conference in 2010 in Austria, based on the yield behavior of different varieties over the whole European region. And this is what I can say in the beginning, totally different what we know, for example, for spring barley. So spring barley, you can grow the same variety in Spain or in Finland. That makes nearly nothing. But for oats, it's totally impossible to do that. And if you can give a more detailed description of these mega environments, you see we have 8 regions that are also overlapping. So this is no clear separation, but there are also overlapping regions. So mainly defined here, the more in the west or in the south you are, the warmer the climate in Europe is, which means here in the southwest you can grow oats in autumn, spring varieties in autumn, including also not only Avena sativa, but also Avena byzantina. We have in Ireland, it's a special situation that mainly spring oats is grown in this country, which has the highest oat yields all over the world due to the statistics of the FAO. We have a true winter oat growing that is limited in Europe to this region, England, northern France, Benelux countries, Western Germany. But I would also say that there's also a winter oat belt, which means also the lowland parts of Austria and Switzerland, which means also the southern parts of Hungary, Croatia, Romania, Bulgaria, and also parts of Turkey. But this is only a belt, which means not northeast of that. You cannot grow winter oats because the winters are too harsh, too continental. And south of it, you have to grow Avena byzantina or spring oats sown in autumn because of this hot and dry climate that starts already in spring. So there's a very, very small belt where you can grow true winter oats. And per definition, we have also the Central European growing area here in the north, and in the far north, the total dependency from day lengths with very early varieties and 24 hours growing of very early varieties in spring cereals. So this is a definition. And if you have a look to what is grown today in Europe, you see that we have oat growing on a very regular basis in many countries. Here, a comparison between 21 and 22. The countries with the highest areas are Spain in the southwest and Poland in the center or in the south in the east, Middle East in Europe. So that means we have a concentrated oat growing not only in the north or in the middle. The oat growing is located everywhere in Europe, which means we have to consider all these aspects of oat growing. So what does it mean? We started to screen for winter damage differences in oats already in 2005, 2006 here at our location, which is very close to Scandinavia, so not a typical place for growing winter oats. But we recognized, even in a quite normal winter to our conditions, so more than 20 years ago already, that there are clear differences. So winter oats, the variety Vista is a very old winter oat variety from German breeding, and true spring oats can also survive here in the beginning of June and can recover And if you look to in the end of March after the winter, you may expect that nothing would survive. But here it survived. So there you can expect that even in spring oats, there's differentiation in terms of cold and frost tolerance. And luckily, we were able also to perform a direct comparison here inside the Oats project. The background of that was not the detection of winter survival of comparison of the yield performance of winter and spring oat varieties, but it was also Fusarium, which was part of the first presentations. So, but we were able here to screen for the differences in a typical winter oat growing region with three varieties of every type for winter and spring sown, sowing in mid of October and beginning of November. And we have recognized that if you have a normal sowing date in the UK in mid-October, so the winter oat has a slightly higher yield compared to the spring oat varieties in this trial without any winter damage. This was a year without winter damage. But if you come to the later sowing date, in generally, the yield decreases, but the spring oat varieties have a higher yield potential. This is 1 year, 1 location, to repeat it. But additionally, you can see, if you have a look to the right side, that the quality in the spring oat varieties have been better in both cases. And additionally, the phenological data, that means here given as the date of panicle emergence, was 14 days earlier in spring oat varieties, which of course can buffer less favorable conditions later in the season, drought, higher temperatures, or something like that by the crop. So this makes, of course, it very promising to say, okay, so spring oat varieties grown in autumn, if they survive the winter, have potential to come to better yield, to better quality, and to higher yield stability. So, and that was my presentation, my introduction, and I would like to hand it now over to Hermann to give the first results of the CROPDIVA experiments.
Hermann, you are muted.
Okay, you see my screen?
Yes.
In full screen or in presentation?
Full.
Full, perfect. Then I go ahead. Yeah, thank you, Steffen. And I now continue with what we have done in 1 year of oat research within the CROPDIVA project. Down. Yeah, it does. So now we speak about a tiny part of CROPDIVA. It's work package 2, and in work package 2 is one of many crops. So it's just oat. And one question that we wanted to zoom a little bit more into is what happens when we grow traditional spring oat in autumn, and we compare autumn and spring oat growing, so autumn growing and spring growing of spring oat over a larger panel and over more locations. So the material that was chosen comprises 250 basically spring oat cultivars that represent much of the diversity in the Central European gene pool. And it has been also chosen because we already have from previous projects that Matthias mentioned marker data on them, like GPS fingerprints. So we can do also genetics on it without investing a lot more in genotyping. The experiments that we set up, we choose 5 locations among the CROPDIVA partners, 1 in Wales, two spots in Germany, 1 in Switzerland, 1 in Austria. And all oat varieties were sown autumn and spring at an appropriate time at the specific location. We grew small yield plots of, depending on the equipment and on the site, about 4 meters square per plot. It was not a fully replicated design, but an augmented design. And we recorded a range of agronomic traits, like the typical ones that we score. We did a winter survival score in spring, and we also used imaging to measure crop cover in autumn and spring with Canopeo. I will say a few words in a minute. Then we took grain yield And we are at the moment measuring grain quality traits on these samples. Canopeo is a smartphone-based app which allows you to take a picture over a plot, and it gives you a soil cover with green plants, and it calculates a value. So we can use this as an alternative to visual scores. It's then a machine type of score, which should be more robust and less biased by the experimentator. The trial sites were Aberystwyth in Wales, Granskewitz in northern Germany, Bad Vilbel in central Germany, which is an organic site, Zurich in Switzerland, and Tulln in Austria. So on each site we had 600 oat plots, 300 for winter and autumn zone and 300 spring zone, always in neighboring experiments. Just by chance, I found a Google Earth image of the oat trial in Tulln, I guess approximately taken in March 2022. And even on the Google Earth image, one can see a nice differentiation in winter survival, as we can see on this image. When we look closer and we go to the plots, then we also can see, similar to what Steffen showed us, nice differences in winter survival. On the right, this is true winter oat as border plots, but in the middle, these are all typical spring oats. We have not done the full data analysis yet, but only partial data analysis, so I can't show you all the results, but we possibly found a few interesting things, which of course we want to look into further, and the same experiments have been planted already this year, so we will have more data next year. What we can see here is we looked at trait correlations between northern Germany and Central Europe in Tulln, Austria, and we have here the Canopeo scores taken in northern Germany, and here the winter survival and Canopeo scores taken in Austria, and we see that they are nicely correlated. So basically, we can get quite reproducible and robust results on winter survival scores. When we look now in the correlation between winter survival and grain yield, we can see there's a moderately positive correlation. So higher winter survival leads by tendency, as expected, to higher yields. And even if we look at the winter survival scores from northern Germany and the yields in Austria, we see approximately the same correlation coefficients, and the correlation coefficients become even higher when we look at the plot yields in Austria and the winter survival scores taken in Austria, so with a correlation coefficient of up to 0.5, as expected, I would say. When we directly compare the yield results between autumn zone and spring zone, I have here the data from 3 locations: Central Germany, Central Europe, basically Austria, and Northern Germany. Then we can see that on all our experimental sites, the grain yield, the kernel yields of autumn zone was better, with a difference of 20 to more than 30% yield advantage of the autumn zone plots relative to the spring zone plots, because this is the 0 line here means no difference between autumn and spring zone, and a bar on the plus side means higher yield of autumn zone, and a bar on the minus side means higher yield in spring zone. And we see this rarely spring zone oat was better in yield than autumn zone. At the moment, we still look at the quality data, the grain quality data of these results. So I can't show you the results on this yet. But if we look a little bit more into trait correlations, this is, for instance, the winter hardiness score, in this case, a visual score taken in autumn, taken in the autumn zone plots taken in spring, and compare it to grain yield. Then we see a nice variation from a score of 3. This means pretty much frost damage, to 9, which means little frost damage or very good winter hardiness. And we see a nice correlation between winter hardiness and grain yield. I haven't taken the example of difference between autumn and spring zone and grain yield, and then the correlation becomes even higher. So we get a correlation coefficient of 0.63 for the difference between spring and autumn and the winter hardiness score. In Tulln, where we had a nice differentiation, as you can see, a score between 3 and 9. This difference kind of disappears. Oh, yep, so okay, and this is the weather data, the temperature data I found for our site in Tulln, and you can see that we had temperatures, particularly mid to end of March, of down to -8 degrees Celsius. So there was not a very severe frost damage, but there was some stress in terms of freezing, longer periods of freezing temperatures down to -8 degrees Celsius. Here is the winter hardiness score taken in Bad Vilbel, central Germany, and grain yield of the autumn zone oat. And here we can see there was little frost damage because scores were only between 6, 7, and 8. And the correlation was virtually 0 between the scores and oat yield. And in northern Germany, we didn't have visual scores, but we had canopeo measurements. And here the correlation between the canopeo measurement and grain yield was again positive. This means again more winter survival indicated by canopeo measurement, and higher grain yield were positively correlated. Yep, this is basically a short glimpse into what we have done so far and the data we have so far in the oat experiments in work package 2 CROPDIVA. It's very preliminary, I know, but we're going ahead with doing more experiments this year, and we still have to go ahead with doing more data analysis on the existing data. And yeah, particularly thanks in the oat project goes to Katrin, who is the project, who is the oat work package leader, but also Matthias, who is work package 2 leader. And I want to thank specifically also Sophie, because Sophie did this preliminary data analysis that I could show you today. Thank you very much. And I think I can stop it and hand over to Selma again.
Okay. So yeah, I will now give a brief summary on our first results on our oat experiments on drought stress tolerance. So as probably everyone here knows, drought is the main environmental factor that can severely limit yield and quality. And with climate change, this is expected to pose an increasing threat. However, information for drought tolerance among oat breeding lines and cultivars is quite limited. Therefore, one of the objectives of the CROPDIVA project is to identify oat lines with improved drought tolerance. And in addition, we aim at detecting drought tolerance associated GBS markers. And for this, a field trial is carried out at 2 locations, in Lleida at IRTA in Spain, and also at the JKI in northern Germany, where the oat panel that is also used in the facultative oat trial is subjected to 2 different conditions, so irrigated versus only rain-fed. And in addition, we are also interested in investigating the effect of cleistogamy on drought stress tolerance, and this is addressed in a greenhouse experiment. And for our field trial, we don't have enough data yet to present any results. So today I will focus on the last point and give an overview of our first results. So in general, during flowering, florets open and anthers are extruded. However, pollination can also occur when the flowers are closed, which is referred to as cleistogamy. And in oats, both flowering types, so cleistogamy and chasmogamy, can be present as shown on this picture here. And in rice, cleistogamy has been shown to protect fertility of flowers from heat stress, which led us to investigate whether clysogamy could also have an effect on drought stress resistance in oats. For this, 50 oat lines from the drought oat field trial that display a high range in clysogamy were selected. And when we talk about clysogamy here, the actual trait that we were looking at is enter retention, because scoring of clysogamy itself is quite challenging, and the number of enters present in the florets after harvest has been shown to be the most affordable and precise indirect trait to score. And the experiment was carried out in the greenhouse, as this allowed better control on the conditions and the timing of the drought treatment. And we had 2 treatments, so well-watered versus water-limited, with 4 replications in a randomized complete block design. And the drought was applied by stopping the water supply for 21 days. And this started when the flag leaf was fully developed through flowering. And the traits that were measured were the yellowing of the flag leaf, plant height, grains per spikelet and grains per panicle, the number of spikelets per panicle, empty seeds per spikelet, 1,000 kernel weight, and enter retention. And all this work was actually mostly done by Ellen Poschel, who did her master's thesis with us. And yeah, she did an excellent job at counting all these things. And this is what our experiment looked like during the drought treatment. So on the left, we see the well-watered plants, and on the right, the water-limited plants. And yeah, so as expected, we found a severe increase of yellowing in the drought treatment, and also significant reduction of plant height in the drought-treated plants, which are both typical symptoms of drought stress. For the enter retention, we found a significant increase under drought, meaning that flowers were more closed. The 1,000 kernel weight was not influenced by drought. However, the number of filled grains was clearly reduced. And next, we looked at the ratio of the number of grains per panicle under drought and watered conditions across the 50 tested oat lines. So everything below 1 means that the number of grains was less under drought, and everything above 1 means that there were more grains in the drought treatment. And this shows that there are actually quite large differences across the oat lines. Some lines were very heavily affected, such as Delphine, and others were not affected at all, for example, Perun. And interestingly, this observation correlated quite well from what we know from previous just knowledge on these oat varieties. And when we look at the relationship of the reduction of number of grains under drought and enter retention, we found quite a small but still significant positive correlation of about 0.3. So this is just a hint that the clysogamy might actually have an influence on the drought tolerance. So to conclude, the correlation suggests that clysogamy might be involved in protecting fertility under drought stress. And a possible explanation could be that pollen and stigma are protected from desiccation, and also that because clysogamy requires less plant resources than chasmogamy, because no energy is needed to open the flowers and extrude the anthers, that more energy is left to develop the grains. Of course, this needs to be validated in further research, and our idea for the next step would be to assess the correlation of enter retention and the reduction of grains in a recombinant inbred line population to reduce possible effects due to differences in the genetic backgrounds of the oat lines. So thanks for the attention. And I will now hand over to Susanne, who will talk about the mixed cropping trials.
You are muted.
Yeah, no, I was busy in sharing the screen. Sorry. Can you see it now? Can you see the screen?
Yes, but
Yes.
it's just the it's the like editing view.
Better now?
No.
It worked before we started because so
It's
maybe
okay.
I have no I stopped sharing and then I do right away here the slideshow. Try that again. Stop sharing. Okay, neue Freigabe. No, it doesn't do at all.
Yeah,
What's
that's good.
that?
It looks good actually. Yeah, you're good.
Oh, I have it now on my other screen. Okay, well, that's good. If it works like that,
Great.
it's perfect. Okay, so hello, everybody. I think this is the last presentation of our webinar today. I will present even more preliminary results than from the oat facultative and the drought experiments. It's about the mixed cropping systems within CROPDIVA. No, I can't continue. I have to close my video and go back to the other screen, otherwise I can't continue. Sorry. I can't see my own screen anymore. Okay. Can you still see it?
No, it's all black.
Ah, Jesus.
If
Sorry.
you have to, you can just use the mode that's, oh, now we're seeing a lovely beach scene.
Yeah.
I want to go there.
But that's it.
Suit.
No, it's not suit. It's used. Come on. No, I'm going
If
to.
necessary, just
This is
show
the
it.
wrong screen. Sorry, I go
It's
back.
also not
Visual
bad.
target. Yeah, I know. Okay, so I try once again. This should be not a screen, not a beach. Can you see that again? Still the beach.
Yeah, we can see it, but it's still not in presentation mode. But if it won't work, you can just try to do it that way too.
Yeah. Okay. Well.
Decide.
Yeah, then I do it like that. Sorry about that. It worked before.
Yeah.
Anyway, so but
And confirm.
Mixed cropping, that's part. Geert, he presented the overview of the work packages, and it belongs to the work package 3 about the diversified cropping systems. And we do there a lot of intercropping and cover cropping experiments, but also, and I'm not going to present that, we're looking at nitrogen and carbon balances, and also have a look about the effect of small landscape elements. And the beauty about CROPDIVA, or one of the beauties, is that it's not isolated by itself, but it is linked also to work package 4, where we will send harvest samples of the best performing combinations and lines to Serbia, where they're specialized on new food and non-food products. And also, Geert mentioned that as well. What? That was too fast. Sorry about that. So where we want to have those lines ending up on the shelf. So Alexander Zon and people in Czechia, they are looking at the development of regional value chains and marketing strategies so that consumers do have something from the work that we are doing there. I'll try again once presentation mode, but it doesn't work for you guys. Okay, so the participants of this work package, or at least of the mixed cropping systems, we have 7 different countries from Belgium, from Spain, Switzerland, UK, Denmark, Serbia, and Germany. And within Germany, we have 2 institutions contributing to that. one of the tasks or the subtasks is that we have multi-year, multi-location experiments, which means that in 3 and 4 different countries, we're choosing the exact same, we chose exact the same genotypes and the same experimental design. So we have winter triticale and faba bean as mixed cropping in Belgium, in Germany, and in Spain, and spring oats and lupins in mixed intercropping in Germany, 2 sites, Serbia and Switzerland.
Sorry, Susanne, sorry, excuse me for a moment. I don't think your slides are advancing.
They're not?
At least they're not for me.
Okay, I go to not share anymore, share again, window.
Something always goes wrong during these presentations, so that's fine.
Yeah.
Oh,
can
there.
you see it now?
Yes, and
okay.
it's full
screen. So you
screen.
didn't see that part, eh?
No, it's fine now.
Okay, good. The overall aim of that is to investigate the feasibility of mixed intercropping of cereals with pulses, and we also look at their nitrogen balances and the nitrogen fixation, because there are big differences from one pulse to the other one. The design is a randomized complete block design. We have 4 blocks, and we have at least 2, but up to 4 different cereal and pulse varieties, and per country 1 to 2 sites. And what we also do is we have treatments with and without nitrogen, and the ratio of the cereal pulses and the seeds per square meter, that depends on the crop and is based on previous experiments. Is it advancing? Can you see a nice drone picture now?
Yes, it's good now. And we have it.
Good. Okay.
It is in presentation mode now too. So that's all great.
Oh, on top of it. Great. Okay. So as I said, like, we don't have a lot of assembled data, but just to give you a few impressions, you see a drone picture. I think that was in Germany. And the experiment started as planned, and we all chose the same experimental design, which is not only the randomized complete block design, but what you see here in the center in yellow is for each treatment. We have 3 plots, and the data we gather, like disease rating and also the harvesting and everything, we only do in the center plot, so we can eliminate border effects. It started as planned, but however, for a couple of partners, the seed supply was a challenge, so we had some delays, and all partly as a result of that, we had in some sites drought or standing water and flooding. We had also in some sites very like high infestation of certain pests and weed infestation. Here, that's in Switzerland, what you see on the top yellow, on the top right, that was a mustard infestation from a previous bee experiment where they allowed the mustard plants to shed seeds. So we had to do a lot of weeding. That's what happens when you do experiments, not on farms, but on experimental site. But we succeeded with a lot of help of civil servants and interns. So that's one of the experiments with the oats and narrow-leafed lupins, and you don't see any mustards anymore later on in the season. So we had to look whether mixed intercropping has an advantage, and yes, for some crop and some variety combination, it did have an advantage. I have later on at least one analysis that we did, and also we were able to define which varieties are matching well, because if you have oats and lupins, they should be harvested at the same time. One should not dominate over the other or have a negative effect on the yield or metabolites that we are looking for. So on top of the multi-local experiments, it was really important for us that we also do regional experiments, like taking into account the demands, the local demands of the stakeholders and the consumers. And there we had like a huge variety of different combinations. We had the various experiments with cereals and pulses, 2 pulses together, cereals and oil seeds, and also undersowing experiments. And there was not only mixed intercropping, but also strip intercropping and couple cropping. And those abbreviations are for Belgium, Switzerland, Germany, Denmark, Spain, Serbia, England, and Denmark. Oh, that was twice. I have to eliminate that for next time. But apart from the fact that we allowed different genotypes, varieties, the design per se was identical, randomized complete block design, and having like a central plot where we do take the data. This is an example from Switzerland where we had mixed cropping of oats, lentils, and Halles barley and lentils. What you see on the left side is an autumn zone experiment with oats and lentils. In the very center, it's a spring zone. And on the right side, where you also see like two little people doing all the work, that was Halles barley and lentils. And we wanted to see also what's the effect of autumn zone versus spring zone. Also for lentils, not only for the oats. They all survived, but I have to admit that we didn't have a real winter in Switzerland last year. For each of the crop, we used 3 to 5 varieties, and we had treatments, as mentioned, also for the multi-local experiments with and without nitrogen. And of course, we had pure and mixed stands to compare that. And before harvest, we looked at the growth stages, the wheat pressure, a couple of diseases, some pests, and we looked also at the lodging resilience and which cultivars matched to each other. And there were huge differences between the latter. Is it still advancing, just to be sure? Hello?
Yes.
Are the slides still advancing?
Yeah.
Can you hear? Yeah. Okay, good, because I didn't hear anything. So although this is a Speaking for Oats webinar, I have some pictures of Halles barley lentils impressions. What you see on the left is the combination of a Halles barley oak ruby with beluga. And they were just perfect. They matured pretty much at the same time. One didn't suppress the other one. And on the right side, we have this Caesar and the green berry lentils. And those lentils, they were so vigorous that they suppressed the barley, and it was lodging by the end of July then when we harvested. So there were extreme differences in terms of other partners compatible with each other. And that's the only data slide that I have. We looked also at the land equivalent ratio. What you see on the left side are all the combinations between the Halles barley varieties and the lentils. And the more green it gets, the better the land equivalent ratio is. And for most of the mixtures, the LER was greater than 1. And the Chateau lentils, like they had like on the very bottom, they had a really nice land equivalent ratio of over 1.5, which is not the case for all of them. The mixture with Goliath, that was even below. So a matching cultivar combination does not mean that the land equivalent ratio is better. So then you have to really define like what's more important for you in terms of a diversified cropping system, and for the grower to be interesting to use that. Yeah, that's it. I'm sorry for the technical difficulties right now, but I'm open to questions if we do have time.
No worries, Susanne. Thank you very much, everybody. Yeah. That was wonderful to hear. One thing, yeah, I'll just add quickly. I'm actually one of those advisory board members for crop.
Yeah.
And I was fortunate enough to be able to go to the last AGM in Quedlinburg. And I must say, I was very, very impressed with this project and everybody and how well they work together. And it's a really big project and there's a lot going on. So I'm sure there'll be a lot of great things coming out of it over the next few years. So I know we are close to being out of time here, but there were some questions in the Q&A here, and some of them have been answered, but I'll just say them out loud for the recording. So Celie Will was wondering, do you happen to know what mega location in Europe maps best to Western Canadian oat growing regions? And Steffen answered this and said, I expect that the northern mega environment in Canada fits well with the Scandinavian oat growing region. Scandinavian oat varieties can also do well in Canada. And yes, that's certainly true. I found it interesting you had 8 mega environments in Europe because there are only 3 in Canada, which of course is much, much larger. So then Sissy Zechner had another question. They drill winter oats by the end of September, beginning of October in Austria, same as for winter barley. So she's wondering, do we already have a feeling for which date would be best for drilling spring oats in autumn for Austria? And Steffen answered this as well and says, typically it should be slightly later sown than winter oats. However, for better winter survival, it should not be sown too late. The optimal sowing window is quite narrow. So that's something for you to think about, Sissy. Hannah Heike had a question about the Fusarium project. Just to clarify, you found around 1% of Langsetiae from the fields in 2021, but 53% of the samples were contaminated with T2. And Charlotte replied and said, yes, while isolating Fusarium, we only found little Langsetiae. However, over 50% of the analyzed samples contained T2, some more and some less. And then Paul Richter had a question. Are there any thoughts that decreased cleistogamy leads to greater outcrossing in oats, or is the pollination already done even though the anthers extrude? And Matthias has replied and said there are nearly no exact data about outcrossing of more chasmogamic types. My impression is that open pollinated types do not show higher outcrossing, so pollination is usually finished. And have I missed anything here? Oh, Hannah would also like to know what was the maximum parts per million, I guess, referring back to the question about T2. So I don't know if Charlotte wants to answer that, or if anyone has any other questions at this time. Have I missed anything?
One.
Go ahead, Matthias. It sounds like you're having a thought.
Yes, Elizabeth Zechner has asked, did you also include naked oats in your studies? Yes, we did. We got 3 lines from Carl Josef Miller. He's also in the audience here. He's also attended. So we looked for these 3 hairless naked oats, and they do also had lower levels of hairs in the Palea and Lemma, isn't it, Selma? They did, if I'm correctly remember.
Yeah, I'm not sure because it was only in the preliminary studies that I looked at them. So I would have to check again, but we didn't include it later because the differences are so big to the other oat lines that we cannot really correlate the trichomes then to differences in Fusarium resistance. So that's why we didn't really continue there. I have to check.
Okay, well, while you're checking that, Jennifer Mitchell-Fetch has another question here. Has any relationship been noted between numbers of trichomes and cleistogamy?
No, we didn't make this correlation, but maybe by chance a correlation. Maybe I'm not sure. We didn't calculate this correlation. We can do it, but we didn't yet.
Lots to do, for sure. Okay, does anyone else have a question for anybody? I don't see anything right now. And it is just 12:30, so we are at the end of our time. Pablo, do you want to add anything? Should I put up the slide for next time?
I would just like to thanks again all because today was such a great webinar session. It was great to hear what's going on in Europe. And a reminder that this webinar is going to be in YouTube, so people can go back and watch them for the next webinar that is will coming. So thank you so much for the attendance and for the speakers again.
Yes, I echo that, of course. Thank you very much. And next time, we don't have the details yet. Once again, my fault. However, Sarah Clark is going to be speaking to us in January at some point. We're not sure exactly when yet. She's with ADAS in the UK. And some of you may also have heard her speak at the International Oat Conference. I actually didn't get to live because there was a glitch for those of us who attended virtually. So I'm very much looking forward to hearing her speak. And with that, I guess we'll say bye for now. And thank you very much, everybody.
Bye. Have a nice
Goodbye.
day or nice evening.
Goodbye.
Bye.
Thank you.
Bye.
Bye.
Thank you
Bye.
so much.
Bye.
Bye-bye.